Good day, everyone. Today, we are going to discuss an important topic in inorganic chemistry, which is errors in measurement. This topic is very important because in chemistry, especially in analysis, we rely heavily on measurements and no measurement is perfectly exact. Understanding errors helps us evaluate how reliable our results are and how much we can trust the data we obtain. Let us start with the basic idea. Whenever we measure something, there is always a difference between the measured value and the true value. This difference is what we call error. In most practical situations, the true value is not exactly known, so we compare our result with a standard or accepted value. Errors are unavoidable, but the goal in scientific work is to minimize them as much as possible and understand their nature. Now, errors are broadly classified into three types: systematic error, random error, and gross errors. Let us first discuss systematic errors. These are also called determinate errors because they occur in a consistent and predictable manner. That means if you repeat the experiment multiple times, the error will occur in the same direction, either always higher or always lower than the true value. Because of this consistency, systematic errors mainly affect the accuracy of the result. Systematic errors can arise from different sources. One common source is instrumental error. This happens when the instrument we are using is faulty or not properly calibrated. For example, if a weighing balance always shows a value that is slightly higher than the actual weight, every measurement will carry that same error. Another source is personal or observational error. This occurs due to the mistakes made by the observer, such as reading a scale incorrectly or making a parallax error while noting the level of liquid in a burette. Then we have method errors, which arise due to limitations in the experimental method itself. For instance, if a reaction does not go to the completion or if there are side reactions, the result will consistently deviate from the true value. Environmental errors are also important, where external factors like temperature, humidity, or air currents affect the measurement. The important point about systematic errors is that they can usually be identified and corrected once the source is known. Next, let us move to random errors, also known as indeterminate errors. These are completely unpredictable and occur due to small uncontrollable variations in experimental conditions. Unlike systematic errors, random errors do not follow any pattern. Sometimes they increase the measured value and sometimes they decrease it. Because of this, they mainly affect the precision of the measurement, which is the reproducibility of the results. Even if you repeat the experiment under the same conditions, you may get slightly different values each time due to random errors. These can arise from slight fluctuations in temperature, pressure, electrical noise in instruments, or even minor variations in technique. Although we cannot completely eliminate random errors, we can minimize their effect by taking multiple readings and calculating the average value. The third type is gross errors. These are the large errors that occur due to the carelessness or human mistakes. Examples include recording the wrong value, spilling a solution, using the wrong reagent, or making calculation errors. Unlike systematic and random errors, gross errors are not inherent to the system but are due to the negligence. Therefore, the best way to deal with gross errors is to avoid them entirely by being careful and attentive during the experiments. Now, let us understand two very important concepts related to errors, which are accuracy and precision. Accuracy refers to how close a measured value is to the true value. If your results are very close to the accepted value, it is said to be accurate. Precision, on the other hand, refers to how close repeated measurements are to each other. If you get very similar results every time you repeat an experiment, your measurements are precise. It is important to understand that a result can be precise but not accurate, meaning the values are consistent but far from the true value, often due to systematic error. Similarly, a result can be accurate on average but not precise if the values are widely scattered. Next, we come to significant figures, which indicate the precision of a measurement. Significant figures include all the digits that are known with certainty plus one uncertain digit. For example, if a value is written as 2.50, it has three significant figures indicating a higher level of precision than 2.5. Understanding significant figures is important because it tells us how reliable a measurement is and helps in reporting results correctly. Now, let us discuss how errors are expressed. One common way is absolute error, which is simply the difference between the measured value and the true value. Since we are concerned with the magnitude of error, we usually take the absolute value. Another way is relative error, which is the ratio of the absolute error to the true value. This gives an idea of how large the error is in comparison to the actual value. When this is multiplied by 100, we get the percentage error, which is often used to express accuracy in a more understandable way. When calculations involve multiple measurements, we need to consider propagation of errors. This means that errors from individual measurements combine and affect the final result. In addition and subtraction, absolute errors are added. In multiplication and division, relative errors are added. This concept is very important because even small errors in individual measurements can lead to a significant error in the final calculated value. To understand the random errors more quantitatively, we use statistical tools such as standard deviation. Standard deviation measures how much the value deviates from the mean. If the standard deviation is small, it means the values are closely clustered around the mean, indicating high precision. If it is large, the values are widely spread, indicating low precision. Random errors often follow a normal or Gaussian distribution where most values are concentrated around the mean and fewer values occur at the extremes. Finally, let us talk about minimizing errors. To obtain reliable results, we should use properly calibrated instruments, maintain stable environmental conditions, follow standard experimental procedures, and take multiple readings. Careful observation and accurate recording are also essential. By understanding the sources and types of error, we can take appropriate steps to reduce them and improve the quality of our results. So, to conclude, errors are an unavoidable part of any experimental work, but they do not make the results useless. Instead, by understanding and analyzing the errors, we can assess the reliability, accuracy, and precision of our measurements and ensure better scientific outcomes. Thank you.